The topic I am going to discuss is immunology. Immunology is the study of defense mechanism of the body. Uh, Important aspects of this topic includes first types of immune system. It is about specific and non-specific defense mechanism. Different forms of non-specific and specific defense mechanism. It includes different lines of defense mechanism like skin glands present in the skin, factors of the saliva, gastric juice. These are external defense mechanism factors, and specific defense mechanism factor. You should learn about it. We should give special emphasis on the structure of immunoglobulin. Ig is the short form of immunoglobulin. It resembles like Y of English alphabet. Different types and mode of functions of immunoglobulin. You must be knowing immunoglobulin are of 5 types. Ig starting from IgG to IgE. Different types of T cells like helper T, killer T, suppressor T, memory T, and various disorders of immune system. Disorders of, especially autoimmune disorders, are generally asked in the competitive exams. You should know also about vaccines. Different first generation vaccine, second generation vaccine, third generation vaccines, and live vaccine or killed vaccine, toxoids, and their examples. Here I am going to discuss about the specific defense mechanism. Two important cells of specific defense mechanism are antigen presenting cell. Antigen presenting cell includes macrophages, which engulfs the pathogen and digest them. These are antigen presenting cells. But the most important cell is lymphocyte. Now, how lymphocytes are formed? Lymphocytes are formed from hemocytoblast, which is a pluripotent stem cell found in bone marrow of adults and liver of fetus. All lymphocytes are ultimately formed in hemocytoblast in bone marrow or liver. After formation, some of the lymphocyte enters into thymus gland, and in thymus gland they further proliferates. Those proliferates in thymus gland are called T lymphocytes. The T word, T alphabet represent thymus gland. The thymus gland for this reason is also called as the training school of T lymphocytes. Those proliferates in the bone marrow are called B lymphocytes. B lymphocytes forms antibody mediated immune system, which is also called as humoral immune system. Humoral. Humor word refers to fluid. If infection has taken place in the fluid of the body like lymph or in blood or in other tissue fluid, the agent of antibody mediated immune system fights with such type of infection. But if the infection spreads up to cellular level, the factors responsible for defense at cellular level is called cell mediated immune system. So we have two types of immune system: antibody mediated immune system and cell mediated immune system. A mode of working of two system: antibody mediated immune system and cell mediated immune system. In this mechanism, what happens? As soon as the antigen enters in our body, it is engulfed by macrophages. Macrophages are antigen presenting cell. When antigen enters into macrophages, macrophages digest them. They break them into fragments. So antigen fragment are formed in the body of macrophages. Then it combines with a complex called Major histocompatibility complex. There are two classes of MSC, MSC1 and MSC2. Antigen fragments binds to MSC2 in antibody mediated immune system. This complex stimulates helper T cells to secrete a glycoprotein, interleukin. Interleukin is also called as cytokine, and it is most important interleukin. It has various category. The most important category of interleukin is IL-2, and its most important source is helper T cells. IL-2 is supposed to be the most important mediator of immune system. Its function is it gives positive feedback to helper T cell. Helper T cell to secrete more IL-2. Macrophages which secretes IL-1 also stimulates helper T cell. So there are three sources from which helper T cells are stimulated. This complex, IL-2 and IL-1. But the most important stimulator of helper T cell is IL-2, and its source is helper T cell. In case of AIDS, the HIV virus, HIV destroy the helper T cell, and so that IL-2 secretion gets stopped or decreased, and because IL-2 is mediated in cell mediated immune system as well as antibody mediated immune system, that's why immunity is decreased. Now helper IL-2 stimulates B lymphocytes to produce two types of cells. Memory B cells, it retains the memory of antigen for future attack. Another cell is called plasma cell, which on division produces 5 different classes of antibody: IgG, A, M, D, E. These are this is on the basis of decreasing number. IgG is most abundant, IgE is least. Of these, the smallest antibody is IgG. The largest antibody is IgM. IgA is also called as secretory antibody because it is present in most of the secretions of the body. It is second most abundant. IgM because it is very large, it's a pentamer, its molecular weight is very large, so it cannot cross the placenta in spite of present in the blood. But IgG can cross the placenta, so it is very good means to provide passive immunity to the fetus, because IgG present in the mother can cross through placenta, and it gives immunity to the fetus. It is dangerous in one case only, in which mother is RS negative and the fetus, growing fetus, is RS positive. It leads to RH incompatibility, a disorder called as erythroblastosis fetalis.